And all right, who's got questions?
Okay, for the gastrointestinal tract, do you need to know like the histology, layers, and all that, or is that going to be more for lab?
So certainly the histology layers are going to be more of a function of lab, or more of a focus in lab, because we are looking at those slides. But because some of that it pertains to the functions of the tract, you do still need to have a grasp on it. So, you know, both from the gross anatomy perspective and from the microscopic anatomy perspective, because we're tying in form and function here, you do need to know that.
Okay. And those are the serosa, right? Muscularis, submucosa, and mucosa.
Yeah, exactly.
Okay. And then all the salivary glands as well, like the name.
Because we didn't really go into like the details, you just need to know the salivary gland functions and saliva and stuff. So we'll worry about the details in lab. They're definitely like, they all have their own like unique secretion ratios and stuff. And so if we had gone into that, then we would need to know it, but
yeah,
we're not going to worry about that.
Okay. And the mouth anatomy, like all the names of everything.
Only the stuff that I put in words on the slides. So if it's just on a diagram, but it's not actually in a slide, then don't worry about it. That's generally speaking, I'm not going to ask you things that I don't actually explain.
Okay. If it's going to be on the exam, do you mind going over like gastric secretions?
Yeah, so definitely the different secretions are relevant. That's why there's slides on them and stuff. Did you have, what specifically was your question about gastric secretions?
How does it occur? So there's neurocontrols, right?
Right. So yeah. So with gastric secretions, kind of your big focus. Hold on. Would you just sit down? Sorry. Okay. Everybody's restless today.
Yeah.
All right. So with gastric secretions, because it's sort of the first part of the tract that's receiving food for chemical digestion, there's some amount of complexity to how the secretions are controlled. So we know that you will start gastric secretion or increasing gastric secretions before the food ever even gets there, okay? So when we split it up into those three phases, the cephalic phase says in anticipation of eating, because your brain knows that food is coming, right? You can see the food, you smell the food. Your stomach prepares for that by increasing secretion so that by the time those food boluses make it into the stomach, there's plenty of gastric juice to mix it up with. So that's what the cephalic phase means. The gastric phase means that while there is food in the stomach, you will have an increase in gastric secretions. And I don't think I put this in there, so you guys don't need to know it, but just if it helps, one of the major factors for that is the pH. So your stomach is monitoring the pH, and if the pH gets too high, so it's not acidic enough, that's one of the things that'll stimulate more secretion.
Okay.
I didn't put that in there, but that is kind of one of the feedback loops, just which I think helps make more sense.
Mm-hmm.
So yeah, so the presence of stuff in there is also going to control it. And, you know, this is also controlling like the motor functions to some extent too. But mostly this is about gastric juice secretion. And then the intestinal phase kind of works the opposite, because now with the intestinal phase, what we're looking at is how is the duodenum handling what the stomach is giving it. So with the intestinal phase, basically what's going on there is if the duodenum feels overwhelmed by what it's receiving from the stomach, it's going to have a negative effect on activity and say, you need to slow it down, you know, so it mostly stimulates it to like tighten up the sphincter, the pyloric sphincter, to reduce or slow down secretion into the stomach. That's kind of the summary of that. As far as what's being secreted, of course, that's going to be a combination. So all of these things are water-based solutions. But with the stomach, of course, it's that combination of hydrochloric acid and pepsinogen. And the other things are important too, but those are the two things that are relevant for the actual gastric juices that are mixing with the food. Because the hydrochloric acid has all of those major important functions, right? It's gonna kill most of the bacteria and other pathogens that come in, so that protects us. It activates the pepsinogen into pepsin, and then it denatures the proteins, so it alters their form to give pepsin better access. And then pepsin starts breaking up those proteins into smaller peptide chains, so that when they end up in the small intestine, there's fewer steps to keep breaking them down into amino acids.
You don't want us to know like the actual length and all that, like of the duodenum, jejunum?
No, you don't need to know numbers.
No, okay. And all the small intestine cell types know their names and what they do.
Mm-hmm. Yeah, exactly.
So the main function of the large intestine is pretty much to remove the water?
Yeah, to remove the remainder of the water. And electrolytes, yeah, but the electrolyte thing is primarily because that's how they drive the water movement. So by pumping sodium out, the water follows.
Okay. But that's pretty much most of it, right? For the
that is most of it. The other functions are all linked to the fermentation of the food residue by bacteria. So that's where we get the vitamins and those volatile fatty acids, VFAs. But yeah, mostly it's just about, everything that goes in there is indigestible, right? We're done with it. We can't directly absorb any more nutrients from it. So we're just pulling out enough water so that when we eliminate the rest of that waste, we're not losing too much water.
In the histology of it, do you like the anal mucosa as stratified squamous and all that, or should we not?
So again, as far as those things having function, you need to know it, but the the details of that will be focusing on in lab. Okay, so Ramon says, what causes gastroesophageal reflux? So you don't need to know that because we didn't talk about it, but basically with reflux, it's circumstances that keep the lower esophageal sphincter too relaxed. And so that allows chyme, if you've eaten, or just gastric juices to splash up into the esophagus. And then the hydrochloric acid is going to damage the esophageal mucosa, cause inflammation and possibly erosion, like eating away at it. And that's going to, obviously cause problems.
Will you be doing the short answer questions?
Yes. So the short answer got dropped off the last exam because it was a lab practical and we really didn't do anything for that part that required critical thinking. But I mean, the course requirements do still have dictate that I have to have short answer. So at least for these lecture exams, yes.
Okay, what do you think that we should focus on? Like, I mean, everything obviously, but that might be for the short answer.
So what I can tell you is that the short answer questions are all going to be in the form of either why does this work this way or how. They're going to be critical thinking questions. So really, I think if you look at the learning objectives, hopefully that will guide your studying in that area. And the other tip is not so much from the studying point of view, but when you encounter the questions on the exam, don't just try to like pause, think, what is this actually asking me? And then answer it. So you want to make sure that you're actually answering the question being asked. Because I get a lot of answers where clearly there was no understanding of what the actual question was, you know, and I get a definition or something. And I'm like, yeah, that's the definition of the thing I'm asking about, but that's not what I was asking you. The other thing is that I've definitely noticed over the last few exams that there are people that are Googling their answers. And so that's the other reason why this is all purely critical thinking. I can't ask you guys anything that can be looked up because it's not always easy for me to tell. And I want to give people the benefit of the doubt, but at the same time, a lot of people are very obvious about their cheating. And so I'd like to discourage that and also make it much easier for me to tell. If I ask you a question that can't be easily Googled, then you either have to answer it by thinking about it and proving to me that you know the material, or you're going to give me a nonsense answer that you're not going to get points for anyways. So that's what we're going for this one. And I will double check the course requirements as far as the final. We'll
the learning objectives are in the, is it in the syllabus maybe, or?
Oh no, so I mean like the ones I put in the lectures.
Okay.
I pull them out of the chapters.
Okay.
Yeah, so it's just kind of like if you're reading the book, each of the sections of the chapters have like the little learning objectives, like this is what we're going to be doing for this part. Yeah. And that's where I'm kind of pulling it out of. So that kind of touches on the overall concepts that you should be getting out of the different subjects or topics that we're actually covering as we go through.
Okay.
So it's going to be definitely more focused on the physiology than the anatomy, because there's really not a lot of critical thinking involved in anatomy, right? It just is or it isn't. But when we get to the physiology, we can focus on like why, right? Like why does it work this way? How would we do it like that? So that's pretty much, I'm just kind of going through looking at the stuff we covered and then turning them into why and how questions.
Okay. Okay, this is off subject from the exam, but do you know the lab simulations, I completed them and it's still showing 0? Or?
Yeah, okay, so the lab simulations are just meant to contribute to your participation points. Because we would have been doing them in lab and you wouldn't have been getting any additional points. When I assigned you the first ones, the way that it was set up, it made it look like I had to give you some points for them. So I set them for a small amount of points. This time around, I realized that 0 was a valid option. So you're not getting points for them in the activity. I'm taking the fact that you completed the activity and I'll be adding it into your participant, like it'll be factored in. Okay. So yeah, so that's why. So as long as you did them, you're good. And I could not find a way to like give you guys access to them repeatedly. I don't know if you can do them again. I could not find, like there was nothing in there that let me give you more than one attempt. So yeah, but those were all very similar to activities that we would have actually done in lab.
Mm-hmm. Okay, so for this exam, we'll have it access to it tonight, and then the same thing until tomorrow.
Mm-hmm. Yeah. Yeah. So this evening at some point, I don't know, 6, 7, I don't know, and then through tomorrow.
Mm-hmm. Okay. And then Wednesday we'll review for the lab exam.
Yep. Wednesday morning we will review for lab exam, and we can start talking about the final exam as well. Yeah. And then we can do another review for that next Monday.
Okay.
Okay, so Ramon says, do we need to know the histology and functions of the accessory organs or just focus on the histology of the stomach? So the histology and function of every part of the tract is valid. So we covered the stomach, and then the second lecture starts with the accessory organs. So it's a slightly different version than what I have on the end of the first lecture because I confused myself. So you do need to know that as well. You need to know the liver and the gallbladder and the pancreas and the histology and functions of the small intestine and stuff too. So don't stop at the stomach. Go through the whole thing. And then there's the metabolism lecture that we did. I did as a makeup as well. So you should have 3 PowerPoints that you're studying off of and 3 recordings. 4 recordings? Recordings, doesn't matter. They're in there. So I know we're getting to the end and everybody's stressed out and there's a lot of material, and on top of that, there's just a lot of other stuff going on, but keep in mind that you made it through the last one, and this is literally 2 chapters. Like that's it. We're covering 2 chapters at the end, and then we will focus on the rest of it. But, you know, compared to the last exam, this isn't a lot of material. So, and I'm not going to ask in any greater detail. So just try to make sure you're going through the whole thing and focusing on the stuff that's on the slides.
All right. I think I'm done. Did you just review today?
Hmm?
Just review.
Just review today. Yeah.
Okay. All right. Well, if anything, can I email you if I have any other questions?
Yep, of course. You can absolutely email me. Anybody else have questions?
Okay, thank you.
You're welcome. If anybody else has questions, now is a good time to ask. If not, email me. Yeah, Ramon, I am grading as fast as I can. It takes time, and I've been trying to get your exam grades back too, so the lab report keeps getting put on the back burner. But I will be definitely focusing on getting all of your grades in over the course of the next week or so, since the final is next week. So I'm sorry that it's taken so long, but there's always been something else. So grading is a slow process. Thanks. All right. Email me if you have any other questions, and I'll see you on Wednesday.